Hello everyone and welcome to the fourth live session of the Cell-to-Cell Metabolic Crosstalk in Physiology and Pathology online conference. This conference has been organized by MDPI and MDPI Cells Journal, and it is chaired by Professor Ciro Isidoro from the Department Health Science, University of Piemonte, Orientale in Italy. I would like to thank everyone, our chair and our conference committee for making the conference possible, and all the participants for joining us today, and for the speakers for giving us their talks. During the month of November and December, we will hold different live sessions, and from the 17th December to the 17th of January, you will be able to read and comment all conference papers presented to the conference. Please check the conference website because the abstract submission is still open, and register for free for the upcoming live sessions. I would like to remind everyone that this webinar is being recorded and that the recording will be posted in the webinar's webpage for everyone to watch it as soon as possible. We will have three speakers in today's sessions. We have rescheduled the talk of Dr. Will Bailis for 2:15 p.m. I will share with you the link for the register because we had some last-minute changes in the program. So the three speakers we'll have today will be Professor Vincenzo Mollace from University of Catanzaro in Italy, Dr. Lorena Urbanelli from Università degli Studi di Perugia in Italy as well, and Dr. Ravi Sahu from Wright State University in Dayton, Ohio, which is in the States. After each speaker presentation, we will hold a Q&A session in which we will address the questions submitted by live online audience. So please, if you're joining us live, type your question at any time during the webinar in the Q&A box. Finally, I would like to introduce our first speaker, Professor Vincenzo Mollace. Dr. Vincenzo Mollace is Professor of Pharmacology at University of Catanzaro and the Director of the Institute of Research for Food Safety and Health at the same university. His major areas of interest are cardiometabolic disorders and nutraceuticals. So please, Dr. Vincenzo, the floor is yours. You can start sharing your screen.
Okay. Good morning, everybody. Thank you very much, Lina. First of all, I would like to thank Professor Ciro Isidoro for the opportunity to be here today, discussing on the role of translocator protein Tspo and intracellular anti-metalloproteinase 2 in dysfunctional myocardial cells in diabetics. So it is known that diabetes is able to produce dysfunction in the myocardial cells, which are two types. One is related to the development of atherosclerosis, and another one is a direct damage in cardiomyocytes, which occurs independently on other risk factors and on the development of atherosclerosis. So in the first part, just to move forward, in summary, you can see that the first part of my presentation will cover a few background aspects which regard the pathophysiology of the so-called diabetic cardiomyopathy. And the second part will focus on altered cell-to-cell connections which occur at the early stages, in particular, of diabetic cardiomyopathy. And finally, we will verify the potential for targeting the mitochondria and especially the Tspo UDAC1 changes in hyperglycemia and how this effect rely with the modulation of the dysregulation of metalloproteinases which occur in the diabetic heart. This is conflict of interest, and from the epidemiological point of view, diabetes represents a consistent risk factor for developing cardiovascular disorders. And you can see that the heart failure in diabetic patients is in the range of 19 to 26%, which represents a quite high incidence in the course of the disease. And in general, the heart failure is higher in diabetic patients compared to non-diabetic patients, and the relative risk is 1.3 for developing heart failure after 46 months. of observation. And it has been demonstrated that in type 2 diabetes, each 1% of the increase of the glycated isoform of hemoglobin is associated with an 8% increase in the risk. And this occurs independently on other risk factors. So the diabetic cardiomyopathy is a specific entity which occurs with the changes over time which appear at the beginning of the disease, which is at the beginning not characterized by loss of cardiomyocytes, but the major target is fibrosis, impairment of the relaxation of the fibers, and cardiac hypertrophy, which at the late stages is accompanied by loss of cardiomyocytes. The pathological changes which occur in the heart of the diabetic patients and in experimental models of diabetes are represented by enhanced thickening of capillary basement membrane. This is occurring mainly in specific districts and tissues, in particular in the retina, in which you have the retinopathy, which is one of the major targets of diabetes, in the kidney, in which you have an impairment of the glomerular functionality, and in the heart, in which there is this thickening of capillary basement. And this is associated to myocellular atrophy and hypertrophy, especially hypertrophy, fibrosis, which occurs as a consequence of impaired regulation of metalloproteinases. And one of the specific components which allows to the definition of the potential role of pathogenetic factor is the deposition of fat droplets. So this represents the scenario which we can expect when evaluating the pathological changes of the diabetic heart. From the pathophysiological point of view, the hyperglycemia, which is accompanying diabetes, and insulin resistance, which is the one which occurs in type 2 diabetes and metabolic syndrome, are mainly accompanied by three key players. The first one is represented by activated renin-angiotensin system, which is tightly connected with mitochondrial dysfunction and impairment of mitochondrial calcium handling, which has been demonstrated occurring in diabetic cardiomyopathy. The other one is the ticket inflammation oxidative stress, which is combined with the microvascular dysfunction we described before. And the other component is the impairment of endoplasmic reticulum stress and cell death, which is associated to the occurrence of advances glycated end products. So overall, these three mechanisms are associated with changes in cardiac stiffness, hypertrophy, and fibrosis, and which at the early stages is responsible for diastolic dysfunction, which characterizes the disease. From the point of view of the leading mechanisms, one of them is represented by the so-called remodeling, which occurs in diabetic heart. Remodeling is a change in a rearrangement of cardiomyocytes, which are the consequence of several factors which are indicated in the panel. You can see that the first step is mainly represented by endothelial dysfunction. So endothelium is the major target for the activities of the early oxidative stress in diabetes. And endothelial dysfunction means the loss of the ability to respond to the injuries through the release of nanomolar concentrations of nitric oxide. The reduction in nitric oxide production and the inhibition mediated by oxidative stress in endothelium, nitric oxide synthase, leads to reduction in cyclic GMP And the reduction in cyclic GMP is a factor which counteracts under normal which leads to cardiac hypertrophy and myocardial increase in myocardial stiffness, and at the end in diastolic and then systolic dysfunction. The other component of this pathway is inflammatory disorder, which mediated by TGF-beta leads to the so-called transition from fibroblasts to myofibroblasts. And this is mainly related to the changes in fibrosis, which are also supported by the inhibition of several metalloproteinases. We especially investigated on metalloproteinase type 2. And in addition, inflammatory disease contributes in the development of each specific change in structural and functional changes occurring in the diabetes. You can see here that all the components which are the leading cause of the development of diabetic cardiomyopathy are accompanied by TLR4 toll-like receptor for receptor which leads to enhancement of NF-κB and then to the release of inflammatory cytokines. The other one is the direct release of TNF-alpha. And finally, we have an overactivation of lipoxygenases, LOX 12 and 15, which are tightly related to mitochondrial dysfunction and oxidative stress. So inflammation is a major characteristic of diabetic cardiomyopathy, and we can define endothelial dysfunction and cardiomyocyte dysfunction as an inflammatory disease. And in addition, we have biochemical changes which are supporting the mitochondrial dysfunction, which is, in our view, one of the major targets for the development of diabetic cardiomyopathy. In particular, the insulin resistance, which are accompanying, as you can see, the type 2 diabetes is connected to an alteration in GLUT4 translocation, which is the transporter which allows glucose to get into the cells. This creates a condition in which glucose is substituted at the early stage by free fatty acids to support energy production in diabetic heart. And you can see that this is mainly mediated by CD36 translocation, which produces this effect. However, the saturation of this mechanism is leading to activation of PPAR-alpha and leads to the accumulation of lipid droplets, which produces at the late stages a dysfunction in ATP production. And these are the changes which can be found in the heart of diabetic subjects with abnormalities in the calcium handling, low ATP production, and then cardiomyocyte death and contractile dysfunction. All those mechanisms are compensatory mechanisms which are occurring at the beginning of the disease and which are saturated at the beginning of the disease. Then we have the apoptotics. And one of the key players we describe is represented by oxidative stress. I'd like to get a little bit into this aspect, since under normal conditions, the accumulation of ROS, which are determinants of the maintenance of several functions, is warranted by antioxidant moieties such as superoxide dismutase and catalase. However, the overproduction of both superoxide anions and the nitric oxide, which is derived by inducible isoformal nitric oxide synthase, lead to micromolar concentration of peroxynitrate, which nitrates superoxide dismutase and catalase and inactivates this enzyme. So we have a defect in antioxidant capacity and an increase at the end stage of ROS and of nitrated species of free radicals. Well, which is the role of mitochondrial dysfunction in diabetic cardiomyopathy? We have seen a series of very specific mechanisms which have been identified. However, we have a few missing links. One is related to the link between free fatty acid oxidation, glucose oxidation, and altered metabolism, but the other one is connected to impaired calcium homeostasis, which seems to be at very early stages of the disease, one of the potential markers to be followed for the development of the disease, but also the potential for the future therapeutic strategies to be arranged at early stage of diabetic cardiomyopathy. So in the last few years, we focused on the potential contribution of metalloproteinase type 2 in its interaction with myocardial oxidative stress. And we can see that this occurs beyond the matrix. So it is known that metalloproteinases may be membrane bound or released and play a role in the rearrangements of extracellular matrix, which is modulated over time by the different activation or inhibition of metalloproteinases. And just a few slides to summarize which are the major roles of these enzymes, which are secreted or may be located on cell surface and produce a degradation of and processing of different substrate. You can see here a list of proteinases, chemoactive factors, growth factors, cell addition molecules, but virtually all structural extracellular matrix proteins are involved in the real assessment related to the secretion of metalloprotein. You can see here that we have a series of isoforms. Today, metalloproteinases are grouped according to the structure in 8 distinct groups. 5 of them are secreted, 3 are membrane bound, and TIMS is a tissue inhibitors of metalloproteinases, which are highly sensitive to the level of free radical species. Well, the other aspect is that the ratio between metalloproteinase type 2 and metalloproteinase 9 is crucial in under normal conditions, but also you can see here in streptozotocin treated animals, and these rely with the accumulation of collagen, which occurs quite early in animals in which hyperglycemia is induced. And this is the effect of this besartan, which is an inhibitor of angiotensin II receptor, and this highlights the role of renin-angiotensin aldosterone system in the development of diabetic cardiomyopathy. So another aspect we evaluated, in which is the link between metalloproteinase 2 rearrangement and the mitochondrium, is, as you can see, the role of the transporter protein Tspo and VUDAC1, which is the voltage-dependent anion channel 1, which are linked and modulated by metalloproteinase 2. Just a few points regarding the role of this transporter in connection with the mitochondrial pole. You can see that Tspo is connected to the channel, which is the driver for anions, and under normal conditions, this is helpful for maintaining the right concentration of cytosolic calcium. You know that cytosolic calcium is crucial for the survival of the cells, and this system is able to buffer the calcium into the mitochondria under basic conditions. And this has been studied in the model of induction of hyperglycemia via the administration of streptozotocin in rats. You can see that after 5 weeks, we have a beautiful increase of glycemia, which is maintained over time for other 4 weeks. And we evaluated the changes in the heart performance in those animals from the beginning to the late stage of the disease. And just a few informations on this slide, which is a lot technical from the cardiological point of view. These are the parameters which are measured by means of echocardiography, and are the ejection fraction, which is a marker of contractility, the shortening fractioning, which is related to the ability of cardiomyocyte to shorten, then to develop the force and tension during the contraction. And then 2 morphological parameters, which are the end diastolic and end systolic volumes, which are other 2 consistent biomarkers. And you can see that during the development at the late stage of the diabetes, we have a reduction in contractility and cardiac damage, which is independent on the development of coronary artery disease. This effect is tightly associated to oxidative stress, but and you can see that the accumulation of superoxide ions, which occurs in the heart of streptozotocin treated diabetic animals, and you can see here that the major biomarker, which is modulated at the early stages of the diabetic cardiomyopathy, is NADPH oxidase. So NADPH oxidase is maybe the driver of the oxidative stress at the early stage of the disease. Well, surprisingly, this effect, of relatively surprisingly, the effect of streptozotocin is not associated at the beginning to apoptotic cell death. If you look at the BCL2 and cytochrome C are relatively unmodified during the development of cardiac dysfunction, that's suggesting that at the early stages, as reported before, we have no cardiomyocyte loss. And which are the changes which are occurring in the calcium buffering through Tspo and VDAC1 in hyperglycemia-related cardiac dysfunction? Just to show, the Tspo is in the huge connection under normal condition with VDAC1 is able to work and to buffer enough calcium in order to reduce the accumulation into the cytosol. However, also mediated through the changes in migration of cytochrome C, under conditions of oxidative stress, we have changes in this machinery which lead to an amplification of the oxidative stress. And this is what we have seen in our animals, in which Tspo and VDAC1 are overexpressed during the development of diabetic cardiomyopathy. And you can see here that both are overexpressed in cardiac tissues. And this effect is accompanied by an increase in cytosolic calcium. And this means that there is a saturation of calcium buffering potential in mitochondria, and that this effect is associated to dysfunction, which is able to amplify the oxidative stress which is occurring in diabetic heart. But the crucial aspect which seems to be relevant in defining the early stages is that there is another metalloproteinase, which is the intracellular truncated isoform of metalloproteinase, which is involved in promoting the induction of specific mitochondrial nuclear stress signaling impairment of cardiomyocyte function and ventricular hypertrophy. And you can see here from this cartoon that we have not absolute changes in the amount of empty intracellular truncated metalloproteinase, but we have a migration from the endoplasmic reticulus to the mitochondria, to the inner layer of mitochondrial membrane, and this effect is also seen that we have disappearance during the treatment of animals with streptozotocin from the sarcoplasmic reticulum into the mitochondria. So just to summarize a little bit, we have that under normal conditions. We have that NADPH oxidase is working efficiently, and the Tspo and BUDAC1 are working efficiently, and we have the right buffering activity against calcium accumulation in the cytosol. And anti-metalloproteinase 2, which is the intracellular isoform, is connected to the MAN and is inactivated. When we have a high glucose and overactivation of NADPH oxidase, we have an increase in superoxide anion, which leads to enhanced Tspo imbalance in the calcium, which accumulates into the cytocol, and this is due or partially due to the migration into the inner layer of mitochondrial membrane of truncated isoform of metalloproteinase type 2. So the answer to the question related to the unmet need is that the imbalance in metalloproteinase 2 and 9 leads to the accumulation of collagen and to the fibrosis. But the early stages are characterized by dysregulation of mitochondrial metalloproteinase, which is able to activate diastolic dysfunction at the early stage of the disease. Well, we are going to the conclusion. This leads to the concept of frail myocardium in diabetic rats, but maybe in many metabolic disorders, the condition of saturation of compensatory mechanism makes the myocardium to be frail against insults. And you can see here that early fibrosis, dysregulation of metalloproteinases, translocation of the truncated isoform, and mitochondrial oxidative stress may be the key mechanisms which are not compensated by antioxidant moieties, which are impaired just from the beginning through nitration of the key enzymes. And which is the relevance of those informations? First of all, it has been demonstrated that preclinical stages, at the early stage of diabetic cardiomyopathy, are reversible. So we can interfere at this stage of the disease in order to avoid that we can move forward in the progression of the disease to the late stages and to the end stage, in which massive cell death, severe hypertrophy and fibrosis, and systolic dysfunction seem to be the characteristic elements which are accompanying diabetic cardiomyopathy. But the other one is that we have the possibility at this stage to interfere with non-pharmacological therapies in order to produce a protection of mitochondrial and to avoid mitochondrial dysfunction. We recently published, this is the result of a working group of the European Society of Cardiology with top-ranking researchers in this working group, which is called Optimal Nutraceutical Supplementation for the treatment of heart failure, and in particular of diabetic cardiomyopathy. And we believe that the contribution of the natural polyphenols. We studied recently polyphenols from the bergamot extract, which is a natural, very rich in antioxidants product at the early stages of diabetes with successful responses. And this means that for the future, we can expect on top, compared to the conventional therapies or previous to the development of diabetic cardiomyopathy to produce beneficial effects targeting the mitochondria and improving the efficiency of this system. And this is just to acknowledge a lot of people which are working in collaboration with us or in our labs. This is my people in my department, but we have a huge collaboration with the group of Rome Tor Vergata and an international board of association of the European Society of Cardiology and the Association for Heart Failure, in particular, and the goals and Maurizio. So thank you very much for your attention.
Thank you very much, Vincenzo, for your talk. Now it's the time for participants to pose questions. So Ciro, Isidoro has a couple of curiosities. You can come in if you want, or you can write it here as you in the chat as you wish.
Vincenzo, that was very nice. Are you thinking that autophagy is blocked and not protecting from cell death because of excess of mitochondrial oxidative ROS production? For instance, anion superoxide is known to inhibit autophagy indeed.
Absolutely. Unfortunately, I had no way to display data, but in the conclusion, you have seen that one of the targets which are characterizing the early stages of diabetic cardiomyopathy is represented by defective protective autophagy. This is what is occurring in our hands. We have no apoptotic cell death, but we have an impairment. If you combine diabetes with other stressors of both sarcoplasmic reticulum and mitochondria, you have the occurrence of defective autophagic response. This is the one of the other components of the frail myocardium in our hands. Okay, thank you.
Yes.
Okay. Now it's, well, participants, they have not posed questions before. Somebody raised her hand, but I think that was an accident. That sometimes happens. So if you have any questions from Vincenzo, you can post it in the Q&A and he can type the answers at any time. So Vincenzo, you can type the answer if you see any question for you.
Okay, absolutely. Thank you so much.
We will move on to our second speaker today, which is Dr. Lorena Urbanelli. She's a researcher and lecturer in biochemistry and molecular biology at the Department of Chemistry, Biology, and Biotechnology of the University of Perugia in Italy. After her graduation in biological sciences at the University of Perugia, she spent her PhD at IRBM, a private research institute in Pomezia, Rome, working on phage display for biomarkers discovery. During her postdoc back at the University of Perugia, she dedicated her studies to the role of lysosomal enzymes as biomarkers of age-related neurodegenerative disorders. In 2008, she became staff scientist at the same university and continued her activity, focusing her interests on lysosomal biology and autophagy in cell models of senescence. Recently, she has extended her interest to the extracellular role of lysosomes and extracellular vesicle release, investigating the effect on lysosomal storage on the release of extracellular vesicles. So, Lorena, the floor is yours. Thank you very much.
Welcome, everybody. Okay, so I'm going to begin. So welcome everybody. Thank you to the organizers for giving me the opportunity to introduce you these topics. And today I'm going to tell you something about extracellular vesicles from endosomal lysosomal system to extracellular signaling. So Let's begin with some recapitulation of some key features of the endosomal lysosomal system. As you can see here in this red spot, the lysosome is the key player of this system. It is round organelles, which is characterized by two main features from a biochemical point of view. It has an acidic intraluminal pH, which is actively maintained by vacuolar ATPases, and it contains a lot of enzymes, hydrolytic enzymes, which are devoted to break down macromolecules into their constituents, which are then transported back, recycled back into the cytoplasm for cell metabolic needs. And in this context, from the beginning, lysosomes were just considered just as a waste bin for the cells. But now it emerged that their degradation process is finally coordinated with the cell metabolic needs. And they are more correctly regarded not only as a terminal degradative compartment, but also as a metabolic hub for the cell, which provides constituents for anabolic or catabolic reactions. But where does the materials to be degraded into lysosome come from? It can come from either outside the cells by different form of endocytosis, or from inside the cell from different forms of autophagy. As for endocytosis, briefly we can recapitulate. We have the formation, there are different forms, of course. It is a complex process of endocytosis. Basically, we have, by simplification, we have an inward invagination of the plasma membrane, which produces an endocytic vesicles. And these endocytic vesicles fuse with early endosomes, and early endosomes later mature or fuse with late endosomes. There are many steps, and not every step is well known. Mature into late endosomes with a progressive acidification of these organelles. And later, late endosomes fuse with lysosome, releasing their content for degradation. And of course, the constituents of this, the macromolecules constituents can then be exported into the cytoplasm and used for cell needs. On the other side, some material to be degraded can come from inside the cell by different forms of autophagy. Briefly, in autophagy, you see there are different forms of autophagy. We will going to recapitulate them later, but you see some cytoplasmic material with this that is engulfed into a double membrane structure, the phagophore. And the phagophore closes and forms an autophagosome, which later fuse with lysosome, delivering to lysosome its content, and the content, of course, is degraded, and the constituents are used for cell needs. But what is interesting is that this process, autophagy, is a process, and is a process that is related not only to the degradation of cytosolic proteins, but also related to the degradation of damaged organelles, such as in this example, damaged mitochondria. And this is very important because in addition to focus the attention on the lysosome, lysosomal degradation as the only man for the cell to dispose of damaged organelles, because there are other processes, other cellular processes which can allow cells to dispose of unnecessary proteins. But autophagy is the only process that allows cells to dispose of damaged organelles. And this is a very relevant aspect. But the endosomal lysosomal system is important not only intracellularly, but we can also say that there is an extracellular side of lysosomes. I mean, this intracellular organelle can undergo fusion with the plasma membrane in a calcium-dependent process that is called lysosomal exocytosis, thus releasing its content outside the cell. Initially, this process was identified in specialized cell types, as for example, in melanocyte for the secretion of melanosomes. But later it emerged that it is a process that can happen to most. It is not known at the moment if all, but in most, for most lysosomes. Two main functions have been associated to this process, to lysosomal exocytosis. And these two main functions are, the first are plasma membrane repair and secretion. The first one, plasma membrane repair, was initially proposed in a seminal paper by Reddy in Cell in 2001. Basically, when plasma membrane is damaged, the cells try to repair it, try to fix the problem by stimulating the secretion of lysosomes and using lysosomal membrane to repair plasma membrane. In addition, of course, when the lysosomes fuse with the plasma membrane, they release their content outside the cell. They release their content extracellularly. And their content mean, for example, a lot of hydrolytic enzymes which are extracellularly not at their optimum pH, which is acidic, from 4.5 to 5.0, but anyway, they can degrade some substrates and they can contribute this secreted lysosomal enzymes to, for example, to extracellular matrix remodeling, which is very important in most cells, but in specialized cell types is also a key process, for example, in bone resorption. As we mentioned, there are different forms of autophagy, but the lysosome is important for all of them. As a matter of fact, we can review here macroautophagy, the three main forms of macroautophagy, microautophagy, and the chaperone-mediated autophagy. These have different features. These are different in macroautophagy, that is so far the best known process, the best characterized process. We have the formation of this double membrane structure, the phagophore, which engulfs cytoplasmic material, damage the organelles and proteins. This structure closed, forming an autophagosome, which fuse with the lysosome, and the material within the autophagosome is degraded. In microautophagy, we have the direct invagination of lysosomal membrane. So lysosomal proteins, specific lysosomal proteins, are engulfed by the invagination of lysosomal membrane. An intraluminal vesicle is formed, and this vesicle is degraded. Chaperone-mediated autophagy is a different process because it does not involve lysosomal membrane. As a matter of fact, we have some cytosolic targets which are recognized by a co-chaperone, which is the shock protein 70, and this complex is targeted to the lysosomal membrane when a receptor, which is the A form of the LAMP2 protein, an isoform of the LAMP2 protein, is able to translocate this complex across the lysosomal membrane for degradation within the lysosome. So these are different processes, but they are characterized by the same key features. I mean, they select some they engulf some cytoplasmic material and then address this material for degradation within lysosomes. Over the last 10 years, another definition involving autophagy has appeared, that is secretory autophagy. So as for the autophagy, so what we mean for secretory autophagy? By investigating some proteins, as for example, interleukin-1 beta, which are proteins that lack a proper signal sequence for secretion, it was discovered that these proteins are secreted outside the cells, but the mechanism is not conventional secretion because they lack a secretion signal, a leader sequence for secretion. It was by investigating this peculiar process, it was discovered that this unconventional secretion of interleukins, of interleukin-1 beta, which exerts its function extracellularly, is mediated by components of the autophagic machinery. And for this reason, the term secretory of autophagy appeared in the scientific literature. The use of the terms secretory autophagy is questioned because according to some authors, autophagy is a process which engulfs cytoplasmic material and addresses it for degradation within lysosomes. But so it would be more proper to talk about secretion dependent on the autophagic machinery. But anyway, we have an important aspect in parallel with the lysosomes. So lysosomal content can be addressed outside the cell by lysosomal exocytosis, and some other content, in the case of interleukin-1 beta, for example, but some other protein can be addressed outside the cell by a secretion process that is mediated by the autophagic machinery. So we have, again, secretion outside the cell in an unconventional manner. Talking about secretion in an unconventional manner, we have to introduce this additional main character of my talk, which are extracellular vesicles. Extracellular vesicles are released outside the cells, and they are released outside the cells in cell culture medium in cultured cells, but they have also been found in every body fluid that has been investigated so far. They have been found in blood and its component, plasma, serum, but they have been found in urine, they have been found in saliva, they have been found in amniotic fluid, cerebrospinal fluid, in milk, and so on. So what are extracellular vesicles, which are another component of the secretory phenotype of cells? Extracellular vesicles are nanometric vesicles, and they are also highly heterogeneous. But anyway, they can be grouped according to their biogenesis in two main groups. And these groups are microvesicles, and exosomes. Microvesicles and exosomes. Microvesicles are the larger vesicles. Their size ranges from 100 nanometers to 1000 nanometers. And they originate from the outward budding of plasma membrane. Instead, exosomes are the smaller vesicles. Usually their size ranges from 30 nanometers to 150 nanometers, and they originate from the endosomal lysosomal system. Briefly, their biogenesis relies on the inward invagination of late endosomal membrane, which produces within the late endosomes intraluminal vesicles. In turn, some late endosomes become full of these intraluminal vesicles and take the name of multivesicular bodies. Multivesicular bodies can undergo two different destinies. They can fuse with the lysosome, forming some poorly characterized structure that are called amphisomes, and so they can be addressed for degradation, but they can also undergo exocytosis, releasing intraluminal vesicles outside the cell. As exosomes originates from this exocytic process, they took the name of exosome for this reason. What is the functional role of extracellular vesicles? Initially, in the 90s, they have been identified in platelets, and they were considered as a tool to dispose of unnecessary or unwanted material during cell differentiation. But now they are considered an additional manner for cell-to-cell communication. So although they have not fully investigated in this context. So our interest in extracellular vesicles in the context of their biogenesis and in the context of the function of the autophagic lysosomal system has been to investigate how the dysfunction of the endosomal lysosomal system affect extracellular vesicles release, or if extracellular vesicles release is affected somehow by lysosomal dysfunction. While lysosomal dysfunction is so interesting for us, is so interesting in general, it is interesting because there are several, more than 40 syndromes, which are genetic syndromes that are characterized by mutation in lysosomal genes or in lysosomal transporter. And this mutation are the cause of lysosomal storage. These disorders of genetic origins are called lysosomal storage disorders. And briefly, because it is a complex process, but briefly, just to recapitulate the pathogenic mechanism, mutation that causes the malfunction of a lysosomal hydrolysis hampers the degradation of the specific substrate of the lysosomal hydrolase. And therefore, this substrate accumulates into the lysosomes. So this is the primary storage. And this accumulation, this storage, that then caused endolysosomal pathway and autophagic pathway alteration, which is the secondary defect. Of course, this pathogenic cascade is very complex, and we are not going to review this cascade, but this is very relevant. It is very relevant also because this storage, this accumulation of undigested substrate within lysosomes, it accumulates lysosomal storage disorders of genetic origin with age-related neurodegenerative disorders, such as, for example, Alzheimer's disease or Parkinson's disease. Here we see some example of genetic disease characterized by lysosomal storage, Niemann-Pick disease with storage of cholesterol and sphingolipids. There are different forms of Niemann-Pick disease, or accumulation of ganglioside in Sandoff and Tay-Sachs diseases. But there are in our hands, we prefer to investigate what happens to extracellular vesicles release during lysosomal storage in pharmacological models of lysosomal storage. As a matter of fact, lysosomal storage can be due not only to genetic alteration, but can be also induced pharmacologically. There are a class of drugs which have a common structure. They are cationic amphiphilic drugs with different therapeutic indication, but a common structure biochemically, with an hydrophobic moiety. You can see here an amiodarone on the left, which is an antiarrhythmic drug, an hydrophobic moiety, and a moiety that can be protonated at acidic pH. So these drugs cross the cell membranes, cross lysosomal membranes, go into the lysosomes and get protonated by the acidic pH of the lysosome. Once they are protonated, they are polar, they cannot be exported from the lysosome, they accumulate into the lysosome and they induce lysosomal dysfunction. For most drugs, like in this example, amiodarone, the accumulated substrate are phospholipids. And this condition induced by amiodarone is called drug-induced phospholipidosis. And this accumulation are peculiar because at electron microscopy, they can be visualized as lamellar bodies, concentric lamellar bodies. For other drugs, actually a minority of them, we have an example on the left is Tyleron, the accumulation, the storage, they accumulate not phospholipids, but they accumulate glycosaminoglycans, so different substrate. The basis for this different accumulation are not fully understood. So what the aim of our work is that the work that I'm going to introduce to you is some results about what happens to extracellular vesicles release during autophagic lysosomal dysfunction induced pharmacologically. We used different drugs, amiodarone and Tyleron, as drugs inducing lysosomal storage of different molecules, as I have already introduced you, but we also use commonly used compound, commonly used experimentally, because they block the autophagic pathway in different manners. Chloroquine is a compound that blocks the fusion of autophagosome with lysosome, and baphyllomycin 1A is instead an inhibitor of the vacuolar ATPases, which are responsible for the maintaining of the acidic pH within the lysosomal lumen. What we did, of course, at the beginning was to develop a cell model who would allow us to test the release of extracellular vesicles upon treatment of cells with different drugs in a relatively easy manner. This model was represented by X cells, which constitutively express the construct M-cherry CD63. CD63 is a tetraspanning, so there are 4 helices crossing the membrane. It's a tetraspanning, is one of the best known markers for extracellular vesicles, enriched in exosome, but also present in microvesicles. So what we did, of course, was to fuse the CD63 with the fluorescent protein M-cherry, and you can see here that if you have this construct, you have a nice membrane localization, whereas if you just transfect M-cherry, you have staining all over the cells. Then, of course, we investigated if these cells really released fluorescent extracellular vesicles. There are different methods to separate, to purify extracellular vesicles. We use perhaps the most used, that is extracellular vesicle separation by differential centrifugation. Briefly, you have a series of centrifugation steps increasing centrifugation speed. Of course, at the beginning, you eliminate cells, then you eliminate cell debris, then you collect large medium extracellular vesicles enriched in microvesicles by centrifugation and 10,000 G, and this fraction is called the 10K pellet, 10K fraction or 10K pellet. And you collect small extracellular vesicles by ultracentrifugation and 100,000 G, and this fraction of enriched in small extracellular vesicles which are richer in exosomes coming from the endosomal lysosomal system is called the 100K fraction. Of course, we detected the expression of cherry CD63 in these vesicles by immunoblotting, and nicely we could measure the AV-associated fluorescence spectrum by fluorimeter measurement, both for the 10K fraction and for the 100K fraction. So using this system, we treated the cells with amiodarone, and we investigate and we try to understand if there were some quantitative changes in the amount of released vesicles. In A and D, you can see the 10K fraction and the 100K fraction, and you can see the measurement of the relative fluorescence associated with vesicles, which clearly indicates that amiodarone treatment induced a dose-dependent increase of extracellular vesicle release for the 10K fraction and to a lesser extent for the 100K fraction. Then we paralleled these results by performing nanoparticle tracking analysis, which is an important technique which allows to count cells, to count extracellular vesicles, and to determine the size distribution of extracellular vesicles. When we count extracellular vesicles by an NTA, both the 10K fraction and the 100K fraction, we obtain a significant decrease of the release of extracellular vesicles of the 10K fraction, not a significant increase for the 100K fraction, and we did not detect any significant size distribution different between treated and treated cells at of course, different concentration and untreated cells. So extracellular vesicles released by cells treated with amiodarone had the same size distribution of extracellular vesicles released by untreated cells. We also checked by electron microscopy from the morphology of these released vesicles, and again, we could not detect any significant morphological change between extracellular vesicles of both fractions released by untreated and treated cells, even if perhaps the extracellular vesicles released by treated cells were more regularly shaped, but no quantitative measurement allowed us to say something significant. We also treated the cells with the other drugs that I have already introduced to you: Tyloron, baphyllomycin A1, and chloroquine. We measured the V-associated fluorescence, and we discovered that all the three drugs were able to increase extracellular vesicles release, mostly of large medium AV. 10K fraction associated fluorescence was increased for all the three drugs. But in the case of baphyllomycin 1A, also clearly of the 100K fraction, whose fluorescence whose fluorescence. Of course, we characterized from the biomarkers point of view our the released vesicles. We did not discover any significant alteration in the biomarkers content upon amiodarone treatment. As for example, ALIX is a marker that better characterized the 100K fraction with respect to the 10K fraction. CD671 was better expressed on the 10K fraction instead of the 100K fraction, but no clear dose-dependent change was observed. At this stage, we decided to investigate the presence of autophagy markers in these vesicles, and we discovered that both in the 10K fraction and the 100K fraction, amiodarone induced an release of extracellular vesicles which are highly enriched in autophagy markers. This is LC3, light chain 3, which is associated with autophagosomal membrane, P62, and BR1. And their presence is clearly increased in extracellular vesicles separated upon amiodarone treatment. We repeated the same experiment with all the three other drugs, and we also discovered that also in this case, when you, of course, you have an increase in cells, but also an increase in extracellular vesicles of the 10K fraction, 100K fraction, 10K fraction, 100K fraction for chloroquine and for baphyllomycin. At this stage, we asked ourselves, if this was some kind of artifact. I mean, you have more LC3 in the cells, perhaps you have more cell debris, and perhaps this cell debris associate with vesicles when you separate vesicles, and therefore we investigated. We tried to assess the localization of LC3 autophagic markers, because if this was just an artifact, LC3 has to be found mostly localized on the extracellular side of vesicles, whereas it could be different if this protein was instead localized within vesicles. So what we did is a protection assay with the proteinase K. Briefly, we separated vesicles, and then in three aliquots. One aliquot was untreated, not treated as a control. One aliquot was just incubated with proteinase K, which can digest everything is located outside vesicles. And one aliquot was digested with proteinase K, but in the presence of a detergent, which is Triton X. And of course, in this case, the vesicles are solubilized, so everything can be digested, located inside or outside. As you can see, CD81 is a membrane protein, it's another tetraspanin. It was not digested, of course, it was present initially in the sample, both in the one, although faintly in the 10K fraction and more clearly in the 100K fraction. It was completely digested by proteinase K treatment. And on the other hand, LC3 that was present initially in the untreated aliquot was instead prevented from digestion by proteinase K. So a membrane protein like CD81 was readily digested by proteinase K, but not LC3 protein. On the other hand, when we used the Triton X, the both proteins were of course digested. These results were consistent with a model, with results were repeated also for the 3 other drugs and were absolutely similar. These results were consistent with a model which is based on the possible release of autophagosomes. I mean, the phagophore is decorated with LC3, then close into an autophagosome. LC3 is also present within the inner membrane. And what can happen is that if this structure is directly released outside, of course, some LC3 is going to decorate the plasma membrane and some is going to remain inside the vesicles, which co-purified with the commonly used method for extracellular vesicles purification. So to take home message, what we obtained by our experiments is to have some indication that the drug-induced lysosomal dysfunction prompted dose-dependent increase in the release of EV, mostly of the large medium size, the so-called 10K fraction, which is enriched in microvesicles, but not exclusively, of course, in microvesicles, but to some extent also of small extracellular vesicles, as in the case of baphyllomycin, which are enriched in exosomes. Both EV fractions are enriched in autophagy markers upon drug treatment, and this enrichment is dependent on lysosomal dysfunction and is not dependent on the type of storage. It can be observed for storage of glycosaminoglycans, for the storage of phospholipids, and so on. So we have evidence that vesicle release from cells in this condition are possibly related to the so-called secretory autophagy, I mean the secretory pathway dependent on the autophagic machinery. Of course, there are many open questions because we don't know at the moment. We are just a few indications at the moment, but we don't know clearly what happens during lysosomal storage disorders of genetic origin, what happened in their released VU. I mean, we don't know if these syndromes, these disorders are associated to the release of extracellular vesicles enriched in autophagy markers. And we also do not know what are the consequences on recipient cells of the release of extracellular vesicles enriched in autophagy markers. An interesting paper has recently appeared in the literature with some indication about this evidence. Because there is evidence by the group of Alessandro Dazzo has provided evidence that murine models of neuraminidase deficiency, which is a neuraminidase is an enzyme that's involved in ganglioside remodeling. So these murine models are characterized by an excessive release of exosomes, and that links this lysosomal deficiency to generalized fibrosis via 2 pathways which are related to exosome release, Wnt pathway and the TGF-beta pathway. But of course, I think that we have still a lot of way in front of us to to shed light on this topic. So I would like to thank Professor Carla Emiliani from the University of Perugia and her group for helping me in communicating you these findings. Thank you, everybody.
Thank you very much, Lorena, for your talk. Now it's time for questions in the Q&A box. Let me see. We don't have any now. Maybe Ciro wants to make some points. I don't know if he's traveling yet.
Just to say thanks very much to Lorena. It was a wonderful lecture. Bye-bye.
So now it should be Dr. Bailis' talk, but we had to reschedule him for later. I will share with you the link for his session that will be at 2:15 today. And then also we will have a break now until Ravi's Sahu presentation that will be at 11:20, so in about 1 hour. And see you later. And thank you very much, Lorena and Ciro, for being here. Vincenzo, he left already. Thanks to him as well. And see you soon. Hello, everyone, and welcome back to today's live session of the Cell-to-Cell Metabolic Crosstalk in Physiology and Pathology online conference. This conference has been organized by MDPI and the MDPI Cells Journal, and it is chaired by Professor Ciro Isidoro. Now the session is about the compartmentalization of cellular signaling, and our next speaker is Dr. Ravi Sahu. Dr. Ravi Sahu completed his PhD from Sanjay Gandhi Postgraduate Institute of Medical Sciences and then pursued his postdoctoral studies from the University of Pittsburgh Medical Center, then Texas Tech University Health Science Center and Indiana University School of Medicine. He's currently working as an assistant professor in the Department of Pharmacology and Toxicology at Bunshoft School of Medicine, Wright State University in Dayton, Ohio. His research interests have been focused on determining the functional significance and mechanisms of pro-oxidative stressors generated oxidized lipid mediator, platelet activating factor, in impacting cancer growth and the efficacy of therapeutic agents, including targeted therapy, as well as define approaches to circumvent platelet activating factor mediated effects. Dr. Ravi Sahu, the floor is yours now. You can start sharing your screen, and to everyone, and welcome back and enjoy the webinar.
So hello everyone, my name is Ravi Sahu, and I would like to thank the organizing committee for this conference for giving me this opportunity to present our work here. So I'll be presenting two different talks today, but they're all interrelated. So first, I'm going to be talking on the lipid signaling pathway in targeted therapy-mediated microvesicle particle release in lung cancer. So lung cancer, as most of us know, is the third most common cause of cancer-related deaths, like worldwide and also in the US, which makes up to 13% of all the types of cancer diagnosed. If you look at its types, its major types, it is of two major types: non-small cell lung cancer, which accounts for the majority of the lung cancer cases, and it further has three subtypes, which are not only different in their prevalence, but also their site of origin. This adenocarcinoma accounts for the majority of these non-small cell lung cancer cases, and it can originate from the cells lining the alveoli. Squamous cell carcinoma, it originates from the flat cells which line the inside of these airways. And this large cell carcinoma, they can begin in any part of the lung. The second type, which is less prevalent, known as small cell lung cancer, it can is been shown, to originate from this bronchi. Even though its prevalence is very less, it is highly aggressive. And if we compare this 5-year survival rate, it is only 6% for small cell lung carcinoma compared to 21% for non-small cell lung cancer. If you look at the common risk factors, it is environmental factors and pollutants such as cigarette smoking and arsenic, asbestos exposure, and also genetic modification. I'll be talking later. So there are so many signaling pathways has been identified that their mutations or hyperactivations contributes to this lung cancer, and if someone has a family history of lung cancer. So this slide basically, tells you that if we divide non-small cell lung cancer by histology, particularly talking about equimoid cell carcinoma and adenocarcinoma, so there are so many signaling pathways have already been identified, that their mutations or amplifications, causes these type of cancer. However, in equimoid cell carcinoma, almost in 20% of the cases and in 40% cases of adenocarcinoma, the etiology is still unknown. So no one knows, what would be, the causal factors, for this type. And if you look at the treatment options, so depending upon the stages of the disease, even though this lung cancer is often diagnosed when they are already in the advanced stage or metastatic stage. But if they are diagnosed in a benign stage, the surgical resection is an option. However, for most advanced cancer types or metastatic types, chemotherapy have been used, for example, platinum-based agents with or without radiotherapy. But now we have FDA-approved targeted therapy, for example, receptor tyrosine kinase inhibitor, and immunotherapy, which is most commonly used, is immune checkpoint inhibitors. And sometimes, we also have a combination therapeutic approaches. So this slide basically shows you one of the various ways that these tumor cells, they develop acquired resistance to these known therapeutic options. In one cartoon here, like as is shown, when the patients are treated with this maximum dose of this therapy, it can kill most of the sensitive cells, but there are still a small population of the resistant cells which can again come back, and there's a term called tumor relapse. However, when these patients are treated with these normal dosing regimens, as you can see, after every treatment, there are also a small population of the sensitive and resistant cells which are still surviving, and they can also can give rise of this tumor relapse. So my lab has been interested in defining the role of a most potent lipid mediator, which is known as platelet activating factor, and its role in cancer growth and cancer therapy efficacy. So if you look at the structure of this path, it is lipid derived, 1-alkyl, 2-acyl, phosphocholin, which acts through a G protein coupled receptor, which is known as PAF receptor, which is widely expressed. It is expressed in granulocytes, B cells, epithelial cells, some mesenchymal cells, and tumor cells. And it is also coupled to many signaling pathways like MAP kinase pathways and so on. And many studies, including ours, have shown that this PAF receptor signaling mediate various pro-inflammatory and chronic pathological conditions, including cancer growth and metastasis. If you look at PAF biosynthesis, it can be synthesized by enzymatic means, which is also known as de novo pathway. And here, what happens is that this cytosolic phospholipase A2 enzyme, what it does, it removes saturated fatty acid, which is arachidonic acid, from this membrane glycerophosphocholin and converts that to inactive lysopaf. Then this lysoacyl transferase converts this inactive lysopaf back into this active PAF, which is then metabolized by PAF acetyl hydrolase enzyme. This arachidonic acid, which is released, it is then acted either by cyclooxygenases producing various prostaglandin, thromboxane, or this lipoxygenases producing heat lipid or these leukotrienes. In addition to this enzymatic pathway, studies including ours have shown that many oxidative stresses, for example, ultraviolet B radiation, cigarette smoking, chemotherapeutic agents, radiation therapy, they all, because of their ability to generate reactive oxygen species, they act on this SN2 position of this membrane glycerophosphocholine and can directly generate this active PAF, as well as many oxidized glycerophosphocholine, which act as PAF-like lipids. And this pathway can be blocked by antioxidant. So as I mentioned, if this PAF is produced acutely, it can mediate pro-inflammatory responses. However, in its delayed response, it induces immunosuppression and carcinogenesis as one of the major focus of my lab. So this is, another cartoon showing you the PAF and PAF receptor signaling. So this binding of PAF to this PAF receptor, which is coupled to GQ, protein, it leads to the catalysis of this phosphoinositol diphosphate through this phospholipase C beta subunit into inositol triphosphate and diacylglycerol. This IP3, they can then stimulate this calcium ion channel to mobilize this calcium ion release. And then, this calcium sensitive cytosolic phospholipase A2 enzyme, which we just know that basically they play, a rate limiting, step in the generation of this inactive lysopath. This calcium ion then binds along with the diacylglycerol and can activate protein kinase C and its translocation to the membrane. If this path binds to the GI, coupled, PAF receptor, it can inhibit adenylase cyclase mediated conversion of ATP to cyclic AMP. And then it's been shown to activate protein kinase A. However, not much has been, studies here. What happens to this, once this signaling gets activated, it can mediate various cellular responses, including leukocyte activation, aggregation of the cells and platelets, ROS generation, angiogenesis, MAP kinase activation, and also induced expression of various, cytokines and chemokines. So now I'll be walking you through to some of the rationale that has supported, this project. So if we talk about this PAF receptor and lung cancer, okay, so we have published a study a couple of years ago that if we take implant this Lewis lung carcinoma cells into the mouse that has expressed PAF receptor and also in PAF receptor knockout mouse and treat them with or without CPAP, which is a known PAF receptor agonist, and measure tumor growth. So here we are looking at tumor volume over the period of time. What you can see here, treatment of this PAF only augments increased growth of tumor into the wild type mouse that has PAF receptor, but not in PAF receptor knockout mouse. And if you measure this lung mets, what you can see is that this wild type treated, mouse with CPAP had increased number of lung mets compared to this knockout mouse. So this study suggests that PAF receptor activation plays a role in lung tumorigenesis. If you look at the rationale from a clinical study, another group have shown that that basically if you take a non-small cell lung cancer tissues of various stages and also the adjacent normal tissues and do PAF receptor staining. So what they have shown is that PAF receptor immunohistochemistry staining increases as these tumor stages progress. So this suggests that PAF receptor is involved in tumor progression. And when they looked at survival probability, what they found is that the patients having high tumoral PAF receptor expression had significantly less survival compared to the patients who have low survival. So that also suggests that this PAF receptor signaling plays a significant role. Now, if you look at the rationale of this MVP in lung cancer, so what this group have done is that they have isolated microvesicle particle from the patient's blood plasma and added back to these HUVEC cells, which is one of the widely used model of studying angiogenesis. And what you are looking at here is that addition of this MVP induces tubule formation in a dose-dependent manner. However, since couple of years ago, it was not known whether or not PAF receptor signaling can affect MVP release in cancer models. So now, like I'll show you some of the studies from our group that puts together the rationale of studying, of looking that these pro-oxidative stresses stimulate microvesicle particle release. So in this study, what our group, has done, has taken this epidermal keratinocyte Hackett cells, which are PAF receptor positive, and treated them with various doses of this UVP. Okay, what they found is that UVP stimulates MVP release in a dose-dependent manner. You know the and and the same effect they can replicate if they treat the cells with the known PAF receptor agonist, which is CPAF. Okay, this MVP release can also be measured in a time-dependent manner. However, interestingly, the like what they found is that this MVP release in response to UVB or CPAF can only occur in PAF receptor positive cells and not in PAF receptor deficient cells. So this is, the finding from our lab, this last year. So what we have shown is that, if we take this PAF receptor positive pancreatic cancer cells, which is PANC1 cells, and also PAF receptor deficient, cell line, which is HS766T, and this KBP and KBM as shown here was used as a positive and negative control. So what, and then if we treat, like these cell lines with chemotherapeutic drug gemcitabine, what you can see is that gemcitabine induces MVP release in PAF receptor positive PANC1, but not in PAF receptor deficient HS766 cells. So that also suggests that PAF receptor expressing tumor cells can secrete MVP release in response to pro-oxidative stresses. However, basically, I'm going to be showing some data on targeted therapy. Okay, so this cartoon shows you FDA approved therapeutic like options for treating various malignancies. And if you look at non-small cell lung cancer, we have gefitinib and erlotinib as a targeted therapy to treat these patients. And the way this therapy work is that they only kill tumor cells which have mutations in the EGFR gene. So the rationale then again comes, from this study, Okan et al. Showing that if, you take this H1650 cell line, which is EGFR wild type, and also they did site-directed mutagenesis to create H1650G cell line and treated them with gefitinib and measured ROS release. So what basically they have shown is that, compared to this EGFR wild type cell line, EGFR mutated cell line significantly generates ROS release. Okay. This is the rationale from second study. What they have shown is that they took A549 cell line, which is EGFR wild type cell line, and they have shown that treatment of erlotinib induces ROS release in a dose-dependent manner. Okay, so these studies suggest that that in addition to their primary mechanism, which is acting on only tumor cells which are mutated, they can also generate ROS from the wild type cells. So now we begin our project, just to first verifying that these targeted therapy act as a pro-oxidative stressors and can generate ROS. So here, so what we see is that, if we take this A549 cell line, which are PAF receptor positive, and treated them with gefitinib and erlotinib, what we saw is that both these targeted therapy generates ROS in a dose-dependent manner. Then we also wanted to verify not only this ROS producing effect, but also the role of PAF receptor signaling in this. So as shown here, this gefitinib erlotinib induces ROS, and we here we use CPAF, which is a PAF receptor agonist, as a positive control, which also induces ROS, and also PAF receptor independent agonist, which is PMA, phosphorylmeristate acetate. They both induce ROS. And targeted therapy and CPAP and PMA induced ROS can be blocked by pre-treating the cells with N-acetylcysteine, which is a known antioxidant. However, if we pre-treat the cells with a known and specific PAF receptor antagonist, which is Webb compound, it can only block gefitinib, erlotinib, and CPAP-induced ROS release, but not from this PMA, which is PAF receptor this independent agonist. So these data suggested that this gefitinib and erlotinib induced generation of ROS was mediated through this PAF receptor signaling. Now we looked at their response on MVP release. So what we did is that we took a cell line, treated them with gefitinib first at various time points, and measured MVP release. What you saw is here is that the CPAP and PMA was used as a positive control. It induces MVP release. However, gefitinib, we saw that at 4-hour time point, it significantly induces MVP release. However, there was no difference between 4, 6, and 8-hour time point, and then its release goes down. So we took this 4-hour time point for our further experiments. Then we tested the dose response effect of this therapy. What we saw is that regardless if you take A549 like cell line or H2199 cell line, gefitinib or erlotinib induces MVP release in a dose-dependent manner. Now we ask a question, that at this particular experimental settings, can these targeted therapy only specifically stimulates MVP release, or they can also generate apoptosis or induce apoptosis? So to answer this question, we treated this A549 cell line with or without gefitinib or erlotinib at 4-hour time point that was used to measure MVP release. And then we quantified apoptosis by measuring caspase 3-7 activity. What we saw is that, at this particular time point, gefitinib and erlotinib do not induce apoptosis. So MVP release was very specific. However, if we go at later like time point, for example, 72 hours, both these agents significantly induce apoptosis, and which can again further be verified by cell survival assay, that both these these agents significantly decrease the cell survival of A549 and H1299 cell line. So now we ask a question, and we wanted to verify the role of PAF receptor signaling. So we ask a question: can blockade of PAF receptor signaling would decrease, significantly attenuate targeted therapy-induced response? So what we did, we treated, the cells, with or without different stressors, and then we took 2 different approaches. So first, knocking down this PAF receptor expression by this siRNA. Okay, so what we saw is that we tested 3 different clones of this siRNA and found that this siRNA 3 clones significantly reduces the expression of endogenous PAF receptor. And then we transfected this cell line with or without scramble control or this siRNA 3. Okay, so as you can see here, CPAF PMA uses a positive control, induces MVP release, similar to as seen, with this targeted therapy. Okay. However, this knocking down of PAF receptor can specifically block CPAF-induced MVP release and gefitinib and erlotinib, but not this PMA, which is PAF receptor independent agonist. So these data suggest that the PAF receptor does play a significant role in targeted therapy-induced MVP release. We then took this second approach where we use PAF receptor antagonists, and what we found is that pre-treatment of the cells with this Webb compound also significantly reduced CPAF and gefitinib and erlotinib, but not PMA-induced MVP release, which again confirmed our finding. So this release of MVP involves, the biogenesis of MVP involves acid sphingomyelinase like pathway. So what it does is that activation of acid sphingomyelinase, it translocates to the membrane, which then cleaves this lipid sphingomyelin into ceramide and then secretes MVP. And this acid sphingomyelinase can be blocked by acid-specific inhibitor imipramine. So what we did is that we took these cells treated with or without these stressors and also with acid sphingomyelinase inhibitor. And what we found is that pre-treatment of the cells with imipramine blocks MVP release regardless of this stimuli use. So that also suggests that this MVP release was specifically mediated by acid sphingomyelinase pathway. This PAF receptor, like we have shown, is coupled to this MAP kinase pathway, and in this this MAP kinase pathway, particularly P38, has been shown like before to play a significant role in stimulating acid sphingomyelinase pathway, and ERK pathway has been shown to mediate PAF receptor induced chemotherapy effect. So we ask a question, that by blocking this MAP kinase pathway, can we attenuate targeted therapy induced effect? So what we did is that we treated these cells with or without ERK or P38 inhibitor followed by treatment with these different agents. And what we saw here is that if we pre-treat the cells with this ERK inhibitor or SB inhibitor, it not only blocks CPAF and targeted therapy induced effect, but also PMA. So now you would wonder, why PMA induced effect? Because PMA, it is PI3 kinase agonist, and that also has a crosstalk with this MAP kinase pathway. Okay. So taken together, here, like what we had shown is that these targeted therapy, which inhibits receptor tyrosine kinase, it has a crosstalk with this PAF receptor signaling, which can be activated, like by this targeted therapy or its agonist CPAF and induces ROS. This PAF receptor, which then also stimulates acid sphingomyelinase to induce MVP release, that can be blocked by its inhibitor, this MAP kinase pathway inhibitor, and also by blocking this PAF receptor either by siRNA or by antagonist approach. So with that, I would like to thank my colleagues and collaborator, Dr. Shripa Chauhan, Dr. Anita Tyagarajan, Dr. Jeffrey Travers, and Yan Fan Chen, and the financial support. And thank you very much for your attention. And I will be happy to take any questions.
Thank you, Ravi, for this nice talk. Do any participants have any questions? If you have them, post them in the Q&A box. Now it's the time.
May I ask a question? Perhaps I missed the point, but if you can add the comment on your opinion about the functional consequences of this increased release of MVP that you find upon lung cancer targeted therapy. Have you can you comment on that?
Yeah, so as I mentioned, like in our future studies, what we are going to do is that we are going to take these MVPs and then mix together with the tumor cells to then implant into the mouse. So basically we would expect that the mouse harboring these tumor cells mixed with these MVPs would show much increase tumor growth compared to the mouse, which would only be implanted with this tumor cells. So that's our expectation based on our data that shows basically this oncogenic PAF receptor signaling can impact this efficacy.
Okay, thank you.
So if there are no more questions from participants, also you can email your questions to cells2020@mdpi.com, and we will forward them to Ravi or to Lorena if you also have some questions from Lorena's talk. And I think we will end the live session here. So before we go, I would like to thank once again Ravi and Lorena and Vincenzo that he already left, sorry. And I hope you enjoyed it. Take care as well. And we will, so Dr. Bailis had to talk before, we had to reschedule him for 2:15 set, so in around 2 hours. So of course, join us, check the webinar's website, the conference website for the registration link there. And that's all for now. Take care and hope you enjoyed it. Bye-bye and thank you.
Thank you, Lena. Thank you very much.
Today, we will have one speaker in this session, which is Dr. Will Bailis from Children's Hospital of Philadelphia and the University of Pennsylvania, and in the Department of Pathology and Laboratory Medicine. After his presentation, we will hold a Q&A session, so please type your questions in the Q&A box at any time during the session, and they will be discussed after Dr. Bailis' presentation. Now I'd like to introduce our speaker, Dr. Will Bailis. He is an assistant professor in the Department of Pathology and Laboratory Medicine at the University of Pennsylvania and the Children's Hospital of Philadelphia. He received his PhD in immunology from the University of Pennsylvania, where he studied the role of notch signaling in helper T-cell differentiation in laboratory of Dr. Warren Peer. He then joined the laboratory of Dr. Richard Flavel at Yale University for his postdoctoral fellowship, where he studied how cellular metabolism controls immune cell functional programming using CRISPR screening. Now, at the Children's Hospital of Philadelphia and the University of Pennsylvania, the Bailis Laboratory aims to understand how the spatial partitioning of metabolism between organelles in cells, among cells in tissues, and across the organ systems of the body controls development and function in the immune system. So thank you, Will, for joining us today and all the participants, and the floor is yours. You can start sharing your screen.
Thank you, Elena. And I just want to start by thanking the conference organizers, Ciro, for inviting me, and Elena, and all the other, everybody else helping put this conference on. It's a real pleasure to be here today. So as Elena said, my lab is at the Children's Hospital of Philadelphia and the Perlman School of Medicine at the University of Pennsylvania. And today I'm going to be talking about some of the work we've done looking at the regulation of T cells by the compartmentalization of this key metabolite, NAD. So before I begin, I just want to take a kind of a bird's eye view of how we at the Bailis Lab approach our research. And like many of you, we're fascinated by this kind of fundamental observation of biology, which is that while all forms of life can start as a single cell with a single genome and divide and give rise to many progeny, all with an identical genetic code, while cells like prokaryotes do so in a manner that gives rise to cells with a very narrow range of functional properties, we know that eukaryotes, and in particular multicellular eukaryotes, are able to do something pretty amazing, which is as they divide and continue to proliferate, they can give rise to many different functional states, so diverse that they're capable of supporting a complex organism like a human being. And this is all done, again, through the same single genetic code. So the way that we classically understand this is through the central dogma and signal transduction. And I'm sure we're mostly familiar here with this, but this is the process in which cells receive internal external information that's relayed to the nucleus to launch a new transcriptome and in turn a new proteome. And we understand this to be the process that drives cells from a starting cell state, here cell state 1, to a new cell state, here cell state 2. But we at the Bailis Lab, while we acknowledge and working upon all of the marvelous years of work showing the role of signal transduction and gene regulation in regulating how cells develop and differentiate, we know that genes and gene regulation aren't unique to multicellular eukaryotes. All orders of life have genes and undergo gene regulation, and we don't think that's necessarily what makes multicellular eukaryotic cellular diversity the only thing that makes it possible. What we're really struck by is all the layers of biochemical partitioning that happen in multicellular eukaryotes. So whereas prokaryotes essentially are just one big bag of a cell where they have all their biochemical molecules in one space, and as they make new proteins from the genetic code, those proteins diffuse and immediately interact with the biochemical environment they're in. Eukaryotes have this really intricate series of spaces they generate inside the cell that we call organelles. And this is true down to the way that our DNA is organized into chromatin domains, that our DNA itself is separated from the rest of the cell by a nuclear envelope. And then again, all the different membrane-bound compartments we have that we call organelles that allow multicellular eukaryotes to partition where and when certain metabolites go, where and when certain proteins go. And that allows them to control the rate at reactions that occur, when they occur, and how they occur. And we and many others believe this is really what gives rise to functional diversity of the eukaryotic cell makeup. And this goes beyond just intracellular partitioning when we're talking about humans.
This biochemical partitioning
we know that
not only
I'm sorry about that. There's some audio footage that's still saved over this. That this biochemical partitioning, it doesn't happen just within cells. We also know that the tissues that make up our organs partition metabolites amongst the cells that make them up. And we know at a very basic level that all the organ systems of our body have unique biochemical roles. For example, our livers store glycogen and release sugar stores to the rest of our body. Our kidneys take up and remove nitrogen and urea from our system. And biochemical partitioning really is just a fundamental way that our systems are organized. So with this in mind, I'm going to be discussing how this type of partitioning helps control helper T cell differentiation in the immune system. So I'm sorry about this. Here we go. So for helper T cells, the way we understand how these cells behave is that they start off in this naive, unactivated state where they wait until they receive signals from their T cell receptor, as well as cytokines in their environment. And when they receive these signals, they begin to proliferate and divide and rapidly epigenetically remodel. And depending on the cytokine environment, these cells will adopt one of several helper T cell states. And the one I'm going to be focusing on today is the helper T cell 1 subset. And these cells are defined by their transcription factor TBET and the production of the cytokine interferon gamma. So when we're thinking about helper T cells differentiating, we're aware that they must meet really unique biochemical demands throughout this process. So helper T cells, as I mentioned before, start out as in cell cycle arrest as a naive T cell. And the process that support this are really just kind of basal mitochondrial respiration to support just arresting cell state. But upon encountering their cognate antigen through the T cell receptor, these cells will begin, as I mentioned, rapidly dividing and epigenetic remodeling within 24 to 48 hours. And then only a few days later, these cells will begin to exit the cell cycle and focus all of their energy on cytokine production. And for this reason, we've begun to understand that there's distinct modes of metabolism that need to support these processes that as these cells are rapidly changing under such a narrow time window. So as I mentioned before, early on, these cells are supported purely by mitochondrial respiration. As they activate, they undergo mitochondrial remodeling and increase their rate of glycolysis in order to generate all the biomass they need to generate nucleotides and epigenetically remodel. And then as they become effector T cells and become much more functional, they become much less reliant on mitochondrial ATP production, and glycolysis is known to really fuel cytokine production and generate all the biomass they need. So this is work that's been done over the past 30 or so years. That's really given us a beautiful picture of how helper T cells behave during activation. But it kind of gives us this very linear binary process in which on one side of the of immune metabolism, we have mitochondria driving oxidative phosphorylation, and that keeps cells in a resting state. And at the other end of the spectrum, we have cytosolic metabolism supported by glycolysis, which is understood to drive effector cell function. However, we know that metabolism isn't a binary process, that it's a dynamic as T cell biology is. And I'm sure we're all used to seeing these crazy metabolic network maps that show all the ways the metabolism and metabolites can flow around a cell. So for this reason, we wanted to kind of step away from that binary mode of looking at metabolism and take a more global view of how metabolism supports T cell development and function. And we undertook a CRISPR library screen targeting the entire mammalian metabolome. So the way that we constructed this library is we generated guide RNAs targeting every gene that encodes a metabolic enzyme or transporter as annotated by the KEGG consortium, as well as including several positive controls and close to 1,000 non-targeting controls. And we introduced this guide library into Cas9 knock-in animals expressing an interferon fluorescent reporter, and introduced these the library such that each cell would be missing a different metabolic gene or transporter. At the end of 5 days, we binned these cells by quartiles of interferon gamma expression, which is that key Th1 lineage defining factor. And we sequenced the guides in each of these quartiles to identify the genes that promoted or impaired helper T cell differentiation, and to figure out the metabolic processes that were behind it. So what we were struck by when we actually step back and look at the results from the screen is, rather than seeing a scatter shot of hits and different pathways, kind of here and there in a random way, we found that we actually lit up entire pathways in sequence, one reaction after the other, really driving home that what we were seeing was identifying genuine metabolic pathways and biochemical processes. So here are just a summary of some of the results we have from central carbon metabolism. And as you can see, that we identified known processes that support helper T cell differentiation, such as the pentose phosphate pathway or 1-carbon metabolism. But what we were struck by is our screen highlighted a process that really hadn't been studied very well before in T cells, the citric acid cycle. So just to kind of reorient all of you on what the citric acid cycle is thought to do in mammalian cells, as many of you may be familiar, that one of the key things the citric acid cycle does is produce several key intermediates that can support multiple biological processes. So the first one that we may be most familiar with is the generation of NADH from NAD, which can be used to fuel complex 1 of the respiration chain, which is used to generate ATP. Intermediates of the citric acid cycle can also fuel the electron transport chain through an alternate entry point, consuming succinate in complex 2 of the electron transport chain. And finally, the intermediate citrate can be exported from the mitochondria into the cytosol, where it can be used to generate acetyl-CoA that is understood to support in mammals all histone acetylation, as well as fatty acid synthesis. So for the sake of time, I'm not going to get into all of the experimental details, but we performed a series of guide and pharmacological studies to validate our findings. And what we found were that complex 2 succinate dehydrogenase, as well as utilization of citrate to make acetyl-CoA, were 2 of the most important functions of the citric acid cycle during T cell differentiation to help them make interferon gamma. So to confirm our pharmacological findings, what we wound up doing was obtaining a knockout animal for the key subunit of complex 2, succinate dehydrogenase subunit C, and got T cells deficient for complex 2 and activated them and acid proliferation and cytokine production at day 5. So what I'm showing you here first is our Cre-positive control from this experiment. And what you can see very clearly is that these cells, using the cell dilution dye, cell trace violet, so each of these kind of distinct populations is cell division, where they've diluted this dye by 2. And on the y-axis is cytokine production for interferon gamma. As you can see, like I said before, these cells begin dividing, and under each division, a greater proportion or frequency of these cells begins making interferon gamma, linking differentiation and function as these cells remodel and divide, until you can see in the final divisions, almost all of the cells over here are making this cytokine. What we were really struck by is when we were looking at, excuse me, this keeps jumping around, looking at the SCHC conditional knockouts, we find that these cells actually divide even more than their Cre-positive controls. And you can see that by more cells having diluted this dye. But what we were struck by is, while the kind of dogma around T cell differentiation is that the more these cells divide, the more they're able to remodel and differentiate, and the better effector T cells they should become. We found that these cells actually produce less cytokine, even though they divided more, showing that when we disrupt mitochondrial metabolism, we can split out differentiation and acquisition of a functional program from terminal function of those cells. And a summary of these results is shown here, with each of these dots being cells from an individual mouse. And you can see overall, these cells divided more, but were less functional than their wild type counterparts. And this wasn't just true for function. We also saw this for the global programming of these cells. So what I'm showing you here again is that as protein expression by flow cytometry for that lineage defining transcription factor, TBET, which is lost in our SDHC knockout compared to their wild type control. And when we perform RNA-seq on these cells, we saw rather globally that many hallmarks of helper T cell differentiation were defective in these cells while they were elevated in their wild type counterparts. So really what this kind of built was a model for us, which was, while classically we understand that rapid proliferation and epigenetic remodeling are directly coupled by signal transduction to terminal function and cytokine production, we found that when we disrupted complex 2 of the citric acid cycle, the electron transport chain, we actually found that we enhanced proliferation. And in the data I didn't show you, we actually enhanced histone acetylation globally, getting these cells to be in a state of sustained proliferation and epigenetic remodeling without ever acquiring the terminal effector function. So we believe this data illustrates how complex 2 actually kind of pumps the brakes on these processes of differentiation that we're defining as proliferation and global epigenetic remodeling, and allows cells to exit this process of differentiation which they need to do in order to acquire the terminal effector function. And this is a process that we won't be able to identify looking purely at signaling, is only revealed when we begin disrupting the biochemistry of these cells, that we can show these two processes are actually distinct rather than one and the same. So this obviously then raises the key question, what components of mitochondrial metabolism that support differentiation, so that's proliferation and epigenetic remodeling, that are being antagonized by complex 2. So we went back and looked at our results of the CRISPR screen and try to figure out what other processes might our hits be participating in rather than just the citric acid cycle. And what we found is that the hits we had in the citric acid cycle were all known to participate in two really important shuttling systems that connect the cytoplasm to the mitochondria. So I won't get into the details of these two loops, as there are a lot of protein and gene names in them, but the net result of these two pathways, one of which is the citrate malate shuttle, is that you move acetyl-CoA from the mitochondria out to the cytoplasm. And for the other shuttling system that's directly connected to this, the malate aspartate shuttles, that moves NAD+ from the mitochondria to the cytoplasm and NADH into the mitochondria, where it can be used to fuel complex 1. So these are two shuttling systems that connect these two metabolic spaces and allow, and this is the main way that these two really essential and fundamental metabolic subunits, acetyl-CoA and NAD, are used or moved between these spaces. So we wanted to see what is the role for these two shuttling systems in helper T cell differentiation. So what we then tested was we designed guide RNAs for every gene that is an enzyme in these shuttles, as well as the transporters that participate in these shuttling systems, delivered these guides to Cas9 positive helper T cells, and then looked at what happened to that key cytokine, interferon gamma, after 5 days of differentiation. And then what you can see here is that using TBET guides as our positive control for this experiment, that each one of these dots represents an individual guide tested in quadruplicate for all these different genes, either participating in the citrate malate shuttle or the malate aspartate shuttle. And what you can see is that pretty much every single guide that we tested results in loss of interferon gamma down to the level of losing the lineage defining transcription factor, really highlighting the importance of the shuttling system. So we next, having seen this, that these shuttling systems were essential for T cell differentiation, we wanted to know what the consequences of disrupting them would be on metabolism, so we could begin working out the biochemical mechanism that explains how they actually help T cells out. So what we first did was we worked with Joshua Rabinowitz's lab at Princeton to perform unbiased metabolomic mass spec on these cells. So we took our Cas9 T cells again, activated them under helper T cell 1 differentiation, delivered a guide RNA against one of the key transporters that participates in the malate aspartate shuttle, and then performed LCMS metabolomic analysis on these cells. So one thing we noticed right away is that when you look at the global NADH, NAD plus ratio, so this kind of gives you a sense of the redox state of the cell, that and the cells that are missing this transporter that is essential for the malate aspartate shuttle, we found, and each of these is an individual guide done in duplicate here for mass spec, we find that cells missing this transporter have a reduced NADH, NAD plus ratio. And even though this shuttling system is one of the main ways these cells move NAD and NADH between the mitochondrial and the cytoplasm, this is actually a pretty striking result that illustrates how important this shuttling system is for maintaining redox balance in these cells. And this is not what you normally see when you disrupt the shuttling system and other cells that use this process. So this really highlighted this is a really active and important process for maintaining metabolism in these cells. And the other key thing we saw is that these cells showed a decrease in this key intermediate of pyrimidine synthesis and carbamoyl L-aspartate. So for those of you that are less familiar of what this process is or what this molecule is, it is part of the backbone of every pyrimidine in de novo pyrimidine synthesis. And what happens is aspartate gets converted to this intermediate and carbamoyl L-aspartate and then is used to build the backbone of the pyrimidine ring and all pyrimidine nucleotides. So having seen this, we kind of wanted to understand how N-carbamoyl aspartate synthesis was defective in these cells and why, because this shuttle in itself shouldn't necessarily disrupt the enzymatic pathway that generates this metabolite. So we decided to focus on this aspect of our metabolomics, that the redox state of these cells was disrupted, suggesting that the shuttle may be really important for moving this metabolite into the mitochondria, NADH. So we next wanted to look at what is the role for this shuttling system and supporting two key metabolic processes, glycolysis and respiration. And we did this using the seahorse assay. So we took Cas9 CD4 T cells and then delivered guides targeting either the transporters that participate in the malate aspartate shuttle or the cytosolic isoform of malate dehydrogenase, MDH1, which is a key participant of the shuttling system. So all three of these genes have no direct impact on mitochondrial respiration unless they're participating in the malate aspartate shuttle. So once again, what I'm showing you here is a summary of our results. Each of these dots represents an individual guide against each gene tested in quadruplicate. And what you can clearly see here is that while the rate of glycolysis was unchanged in cells expressing guides against these genes, mitochondrial respiration was reduced regardless of we targeted either transporter, but really strikingly, when we target the cytosolic version of malate dehydrogenase, MDH1, which does not participate in the citric acid cycle to fuel respiration, it can only fuel respiration if it's part of the malate aspartate shuttle, we saw that this too resulted in a decrease in respiration, really driving home the point that these cells use the malate aspartate shuttle to fuel respiration. So our hypothesis from this point forward became that the malate aspartate shuttle, as I said, brings NADH into the mitochondria to support complex 1 and respiration. And in work that was done a few years before, we found this by Matthew von der Heyden's lab and David Sabatini's lab. They had found that in cancer cells, complex 1 was important for supporting aspartate synthesis. And we hypothesized that this is probably happening in our primary T cells too. So we wanted to directly test this. And to do so, we returned to our mass spec system. So we took wild type CD4 T cells, differentiated them for 4 days. And then for just 4 hours, we put them into media that had a complex 1 inhibitor, rotenone, and then sent these cells off to analysis by the Rabinowitz lab at Princeton. And showing us that the drug works. So this drug blocks complex 1, which consumes NADH. So we flip the NADH NAD plus ratio, since there's the mitochondria cannot consume any DH anymore. And this also blocks ATP production in these cells, as would be expected if you block respiration. But consistent with our hypothesis, what we also found is that in day 4 activated T cells, when you block complex 1, you lose aspartate synthesis and the downstream production of the metabolite N-carbonyl aspartate, really consistent with what we were thinking was going on in these cells. So having seen this, we next hypothesized that perhaps what the malate aspartate shuttle was doing and why it's so important in helper T cell activation is because it's fueling complex 1 to make aspartate. And since aspartate is required for all de novo pyrimidine production, and these cells are dividing a lot, which means they need lots of nucleotides, that perhaps what the malate aspartate shuttle and complex 1 are doing is fueling T cell proliferation by helping them generate nucleotides. So we wanted to test this directly. And the way we chose to do this was a rather simple add-back experiment. We took T cells labeled with a cell dilution dye to track proliferation. And then 2 days after activation, we treated them for just 12 hours with a complex 1 inhibitor, and then added back aspartate to these cells to see if aspartate would be sufficient to rescue division. And this is data summarizing these results here. And what you can see is under our control condition with the vehicle-controlled DMSO, these cells are rapidly dividing at this time point. If we simply treat for a short period of time with rotenone, these cells, I'm not showing this here, are all fully viable, but they're just not dividing. And simply by adding back the amino acid aspartate, we can largely recover almost all the divisions that were blocked by rotenone treatment. So not only does this highlight that the key role for complex 1 and the respiration chain at this early time point in T cells is to generate aspartate to fuel nucleotide synthesis, but that the ATP production supplied by the mitochondria is not what it's the primary role of it at this time point. It really is there to help fuel nucleotide production. So to summarize everything I just shown you so far in the talk, that what we believe these two findings really illustrate are the key ways that under this really narrow window, this 5-day window, as these cells go from these resting cells to these rapidly dividing cells, to these resting cells again that are making tons of cytokine and protein, that they need to rewire their metabolism very rapidly in distinct ways to support each of these unique stages. And that early on, the malate aspartate shuttle and complex 1 of the respiration chain helps these cells generate nucleotide intermediates, as well as intermediates needed for epigenetic remodeling, and data I didn't have time to show you, that help this process of differentiation, which is division and gene remodeling. But as these cells start finishing the activation process, complex 2 starts pumping the brakes on the malate aspartate shuttle, and actually, in data I'm not showing you, antagonizes these shuttling systems, helping bring these cells out of the cell cycle and stop remodeling, and enforcing terminal differentiation and promoting cytokine production. When we disrupt these transporters, what we find is these cells divide poorly and epigenetically remodel poorly and kind of growth arrest. Whereas in contrast, when we block complex 2, these cells hyper remodel, hyper divide, and actually never stop doing that process. And in turn, since they don't exit the process of differentiation, they never properly acquire their terminal T cell function. And we really think that together, this illustrates how T cell activation and differentiation of function, which are coordinated all together by signaling, are actually supported by distinct metabolic processes that allow these cells to properly function and change under such a narrow time window. So just to conclude, I want to thank the people that help do this work. Some of this work was done during my postdoc in Richard Fell's lab, along with Justin Shire and Jun Zhao, who helped analyze that CRISPR screen, as well with Yuval Kluger's lab and Joe Kraft. Some of the work was as well done at my lab at University of Pennsylvania and the Children's Hospital of Philadelphia. And I'd like to thank all my lab members, our collaborators, Josh Rabinowitz and Louis Jim Mayer at the Mayo Clinic for giving us the SDHC knockout mice, as well as all my funding. And I'll stop there and take any questions with the time remaining.
Thank you, Will, for your presentation. So now it's the time for participants to pose your questions in the Q&A box, if there are any. If not, you can always email your questions to cells 2020@mdpa.com, and we will forward them to Dr. Bailis. And if there's no questions, which seems not, we will end the session here. So thank you, Dr. Bailis, for accepting to give a talk and for accommodating at the end and rescheduling the talk and being available for that. And well, take care, see you soon. And please, all of you, remember to add Ciro. Do you want to say a comment or anything?
Just say thanks very much to you, Elena, and to
Will, of course. I think it's being cut. So for that, you remember to register for the upcoming live session. The next one is on the 25th. So it's 2 days from now, on Wednesday. And then in December, there will be 2 more, in the 17th and 18th. So see you soon and take care. Bye-bye and thank you.
Thank you.